Hello children. In the previous class we have started to describe the antigens and antibodies. We have seen various types of antigen and let us enter into the structure of antibodies. Structure of antibodies. Antibodies are chemically immunoglobulins. Immunoglobulins. In short, we call them Ig. Immunoglobulins are glycoproteins. They are made up of amino acids and carbohydrates. So that we call them glycoproteins. So antibodies are also called as immunoglobulins. Immunoglobulins are made up of carbohydrates and amino acids, so that they are said to be the example for glycoproteins. Right. Let us see the structure of antibodies. Antibodies are in the shape of English alphabet Y. Actually, they are Y-shaped structures. They are in the shape of English alphabet Y. There are 4 polypeptide chains in the antibodies. Antibodies are composed of 4 polypeptide chains. Of them, 2 are said to be L chains. L chains. 2 are said to be H chains. L for light, L-I-G-H-T, light chains, H for heavy chains. So, each antibody is made up of 4 polypeptide chains. Among the 4, 2 are said to be L chains because their molecular weight is only 25,000 daltons. 25,000 daltons. Probably they have 211 amino acids. Whereas heavy chains are little bit longer than the light chains. Their molecular weight is 50,000 daltons. 50,000 daltons. Number of amino acids present in the heavy chains are 450. 450. So that's the difference between light chain and the heavy chain. Light chains are short in nature, heavy chains are longer. They have 2 compartments. Each light chain is attached with a heavy chain by disulfide bond, SS bond. We call it as disulfide bond. Disulfide bond. Each light chain is combined or attached with a heavy chain by disulfide bond. Both of the heavy chains are attached with each other. This is a flexible hinge joint. Hinge means weak, flexible joint, but these joints are very strong joints. Right? Nothing special to add with the structure. These light chains in all the antibodies are made up of either lambda particles or kappa particles. There is no other particle can be found in the light chains. Light chains are made up of either this lambda or kappa particles, but we may not find both lambda and kappa here. But the heavy chains are made up of 5 different types of particles. Based on the type of particle present in the heavy chain, we classify the antibodies into 5 types. There are IgA antibodies, IgE antibodies, IgG antibodies, IgM antibodies, IgD antibodies. This A for alpha particles. E for epsilon particles, G for gamma particles, M for mu particles, D for delta particles. So, once again I tell you, regarding the light chains, there is no variation among the antibodies because all the antibodies may contain either lambda particles or kappa particles in their light chains. But heavy chains are varying, they may have different types of particles. If the heavy chains are made up of alpha particles, the antibody is called IgA antibody. If the heavy chains are made up of epsilon particles, the antibody is called IgE antibody. Likewise, based on the type of particles which present in the heavy chain, the antibodies are classified into 5 types. Are you clear? I hope you are clear. So, this is one segment. Apart from this, the antibodies have 2 ends. There are 2 ends for the antibodies. This end, the upper end of each chain is called N-amino end. N-amino end. The lower end of each chain is called C or carboxyl end. Carboxyl end. Right? Each chain has 2 ends. The upper end is called N-amino end. The lower end is called carboxyl end. This N-amino end is containing varying amino acids. The type of amino acids present in the N-amino end is changeable. We may not have a constant amino acid in the N-amino end. So that this region, this N-amino end is called variable region. Variable region. Why? Why do we call them variable region? Because the amino acids found here are not constant. They are keeping on changing. But the amino acids present in the lower end or carboxyl end is constant. They are permanent. So that this carboxyl end is also called as constant region. Constant region. Constant region. So the upper end of each chain is called N-amino end. Lower end is called carboxyl end. The N-amino end has no permanent antibodies. The antibodies found over here are changeable, but the amino acids present here are unchangeable. They are permanent. So that this region is called variable region. This region is called constant region. Since the amino acids are supposed to vary, they can be able to identify different types of antigen and destroy them. With this we conclude the structure of antibodies. Thank you. Thanks a lot.